your coffee is also ready with extra shot. Okay, good morning everyone. Assalamualaikum warahmatullahi wabarakatuh. Okay, wow, such a very enthusiastic faces. Yeah, we have approximately, I think according to the capacity of the room, about 150 seats, more or less. Yeah, and these are the first grade, sir. So just to give you some background of these students, most are the first grade, and we also have.
That's the first year?
First year, yes. I mean like first year.
Freshman?
Freshman, yeah.
You look very fresh to me.
I'm entrusting you to burn their spirit, you know, to not regret their decision to come here. Yeah, I know that you can do a very good job on, come on, this is. And we also joined by some exchange student. We have some Malay students, Resan, yeah, also from the Philippines, also some Philippines. So we are quite international today. So thank you again. I know that many of you have not known me yet because I don't teach the first year. My name is Yoga, the meditation yoga. I always introduce myself like that. Yes, I'm one of the staff from the Department of Food Technology, and then I will be the MC. I'm not going to speak, I'm just opening and then give the microphone later on to our young, bright lecturer who will be your moderator. Yeah, that will be Mr. Haider here. But among us, we have an esteemed speaker, esteemed guest coming all the way from Belgium. Yeah, and he is Professor Jan van Impe. Is that correct, right? Jan van Impe. Very well known in his field, and he is today thank you so much, Jan, that he will share with you some of his research. Not only research, this has been like culmination of decades of research. It's not like only 1-2 years. I believe this 2-3 decades possibly. So this is like the essence, what you learned or what you got from very long period of research. So this is like, you know, cream of the crop, if you know what I mean, right? And before, of course, I will hand this microphone to the real moderator and eventually to our real speaker. We also very pleased today that our Dean, Professor Sukiharto, on my right side, that he will give a short speech, small speech, and then maybe to give us an opening to officially open the guest lecture today. So, Professor Sukiharto, the floor is yours, Pak.
Very short. Very short. Thank you, Yoga. Not evening. Good morning, everybody. Morning. Good morning. Yeah. I wake up around 2 o'clock this morning, so it's still like evening. Because my wife is not here, so I have to prepare everything by myself. Good morning, and also Monica, our honorable speaker, especially Jan from KU Leuven, Belgium, and all of you, hopefully you are fine today. Thank you very much, Pak Yoga and Pak Haidir, who has arranged this very good lectures this morning. I myself, I know Jan and Monica since last year. They are so kind and they are so funny. Jan is very funny, so you have to be ready to always open your mouth. And this is also a good chance for our exchange student. We have from Malaysia and also from Philippines. And all of you can speak English, right?
Of course.
Of course.
Yes.
Yeah. If you don't understand, then you can say, "Can I ask in Javanese?" It's okay. Jan understand very much in Javanese, so don't worry about that. I'm so sorry because after this I have to go to Jepara. We have a official meeting in Jepara until tomorrow. And once again, thank you, Jan and Monica, for having with us this morning. We are very, very much wants to learn about everything about the food tech, the most advanced knowledge and research in Europe, especially. I promise to you very brief and overall thanks and have a good lectures this morning until dawn, yeah? Until tomorrow.
You come and stop me.
Okay, anyway, thank you very much and have a very good lectures this morning. Thank you, Pak Yoga.
Okay, I know that you really want to have the lecture as fast as possible, but let's start with a group photo because the dean is leaving. Yeah, can I invite our photographer to come forward, Ms. Basiti? Yes, please. So let's have a two.
You have a wide angle?
I don't know. Let him try. Ini bisa dimatikan sebentar nggak? Apa namanya? Di close aja kalau nggak di close. Okay. Two photos. One photo from here, and then we will move. They will take it from here. Yeah. Invite Monica and Haidir as well, please, to come forward. Mbak, come on. Yeah, from here. Come closer. Careful, careful.
The group is us. I thought the class. Huh? I want a picture of the class.
Yeah, after this we will go there.
Okay. Okay.
3, 2, 1.
Okay.
I stay here? No?
Only this time student can ask Dean to move.
Okay, 3, 2, 1. 3, 2, 1. Again, 3, 2, 1.
Okay, then can we please move there? And use your widest angle either with the phone or with the. Oh yeah, can you please all stand, please? Yeah.
Kelihatan.
HP yang white cuma apa? Berapa kali? Nanti di blog. Oh, panorama bisa nggak?
Waduh, rawan nih.
Ya, tapi berapa?
Anu aja, video.
Can you then take one with mine?
Oh ya.
Huh?
Bagaimana? Enjoy.
That will be our young, handsome, bright, young staff, faculty member. No, not you. Okay, okay. And there will be Mr. Mas Agus Haidir Tamimi. I invite you to come forward, please. Yes, and then I formally resign myself, and now this whole session is on your good hands. Can we have a big applause to Pak Haidir?
Well, okay. Thank you very much, Mr. Yoga. So, again, good morning, everyone. Yeah, so it's a truly pleasure to meet you. It's an honor to be here today. You know, Professor Yan, I still remember the first time we met. I think it was last year. We had this event ISFA. Yeah, you know, at that time I could only hope that one day I might have a chance to stand with you in the same stage. And you know, today I feel very fortunate to share the stage with you. And today, yeah, as the privilege to be a moderator for the sessions. So, for everyone wondering, Professor Jan van Impe is a very respected professor from KU Leuven, Belgium, okay, with strong expertise in predictive microbiology and food process modeling. So he has shared his knowledge through many important research works and collaboration in the field of food science. If I'm not mistaken, today Professor Jan will share us about the positive and negative microbiology and novel technologies. So, well, I'm not an expert in microbiology, but I suppose this topic will cover how new technology can help us, you know, in the positive way, microbes like for food, health, and also biotechnology, and at the same time, how can we handle the harmful microbes such as, you know, may cause disease or spoilage, isn't it? Yeah. So at the end of the talk, as previously mentioned by Mr. Yoga, you know, these are our top students. Yeah, they are our best, and also we have also deep or our exchange student from Philippines and Malaysia, and yeah, of course there will be after the session there will be Q&A, and I suppose we will have many thoughtful and interesting questions. Yeah. So without further ado, please everyone give a very great applause to Professor Yan, please.
Thank you very much for all the introductions. What do I need to do here?
Make yourself as comfortable as
Yeah?
possible, sir.
This is on. They are not going to
like the history. I think we have been 60 or 70 years only.
That's about 70, you said. That's about my age. Yeah, good. Okay. We are making great progress, I can see that. This is my team. It's a big team. Dr. Monica is here. Is she in the picture? Ah, yes, she is sitting there. Now, in the team we do a lot of things. I have prepared a little video. I hope it will go through in the broadcast as well. That summarizes in 5-6 minutes what we are doing in the team. Okay? This video we created at the occasion of existing 30 years. If at that time I will close my eyes, if you then want to leave the room, that's fine with me. Then you know the essence. I'm always serious, as you can see. Okay. And then I will come to, do we have a pointer or something? No. No, that's okay, that's okay. I better sit. Coming to what we do in the team is experimental work in the laboratory and computational work on the PC. Okay? Since we do that in the same team, we can exchange information from experimental results, do mathematical modeling, do data processing on the experiments that we conduct, improve our experiments, and so on. The main objective of my team is to can I put that as yes, is to obtain better technology and better control of processes, so that we keep the fermentations better under control, that we fight the pathogens, that we have healthy food, safe food, and that we can secure food for everybody. That's even the most important one. So this is the team, and if all, well, this was then the updated matrix of our research. You see the number of application domains. A bit more boring are the research areas. We do system theory, we do mathematical modeling. You all like math. I smell the enthusiasm, it's overwhelming. Wow. Who knows what is a differential equation? That weird thing that you try to avoid to solve. Yeah, that's the one. Yeah? Linear algebra, your favorite? Statistics even. Yeah? We have a question online. Ah no, she's gone. You have a good background in mathematics? No? Okay. Good to know. Monitoring, measuring what happens in the processes, optimization of processes, and this we do in a number of applications, chemical and biological reactors. We do waste treatment systems, going from waste to useful products. At the end of the day, waste is an overrated concept. There is no waste. Everything is useful. We need to find the technology, and that includes the microorganisms, to convert a waste into energy, gas, ethanol, what gas? What gas do we produce out of microorganisms? Methane gas, anaerobic digestion, or useful products, new fuels. I will give an example later on. Sustainable aviation fuels, as an example. And of course, we are very active in the domain of public health, predictive microbiology. How can we keep the food safe? That has to do with safety and, of course, also spoilage. A video, I hope it works.
Does it have sound?
It has sound, yes, but we cannot hear it. Should I stop here and do something, or we just let it play? No, that was not wise.
Could you turn on the volume?
You mean here? There's a microphone.
you can just play now. Just play now. Yeah. Just play. Yeah.
KU Leuven, the University of Leuven, is subdivided in 3 major groups. We belong to the group SET, which stands for Science, Engineering and Technology. In that group, we have 5 faculty. I have the echo here, sorry. Faculties, the Biotech Plus group belongs to the Faculty of Engineering Technology. We are based on the campus in Ghent. The Biotech Plus group, I started it in the academic year 93-94, right after my PhD. This means that today we exist exactly 30 years. As professor at KU Leuven, active in 3 domains, this is education, research, and service to society. As far as research is concerned, the Biotech Plus group focuses on the optimization and control of chemical and biological processes. It's a very broad domain that we have adopted a systematic perspective on every part of research that we do. Systems theory thinking is really the core of all of our research. But a multi-scale approach, we research at the level of the smallest entities, that would be chemical reactions, that could be microorganisms, up to the process scale, that would be full reactors, chemical reactors, and biological reactors. At present, Biotech Plus consists of 2 permanent staff members, 5 postdocs, and a lot of PhD students active in many disciplines. Unique in the research organization of Biotech Plus is that we organize our activities in 2 parallel wings. One side of the lab is focusing on laboratory experiments. We have a very well-equipped safety level L2 microbiology lab where we perform a variety of experimental work with many different microorganisms and technologies. The other side of the lab focuses on in silico work. That means we do a lot of simulations, we build mathematical models that summarize the knowledge that we try to gather in the laboratory. In parallel, we design novel experiments with these models so that we can be more efficient in the lab. The results are mathematical models that we can use for process optimization and control at full scale in many applications in food, feed, and pharma. The Biotech Plus group is a large team. That means that on an almost daily basis, we need to look for funding. Since the basic funding from the Flemish universities is decreasing over time, we need to apply for mainly competitive funding with external funding agencies. And this we do at different levels. We apply for projects and grants with our own research council, the Research Council of KU Leuven. We apply for mandates and projects at the Flemish level. And here we have 2 agencies we are mainly applying with. This is the FWO, the Fund for Scientific Research Flanders, and the VIO Agency for Innovation and Entrepreneurship. We also apply for many European projects. Today we are involved in 5 research networks, 2 as coordinator, 3 as partner. In all of these projects there is many input from industry companies involved. I give a few examples. At present we collaborate with Johnson & Johnson, represented in Belgium by Johnson Pharma. We collaborate with Unilever, we collaborate with Danone, Procter & Gamble, and so on.
In the lab, we investigate on the one hand how we can kill undesirable microbes such as pathogens. On the other hand, we investigate how we can use useful microorganisms to have them do specific processes for us. To kill microbes, we are currently developing nanoproteins. The idea is to develop these nanoproteins in such a way that they provide a surface with a self-disinfecting property. That way, we do not have to use any chemicals anymore to disinfect these surfaces. We can use microorganisms for all types of useful processes. For example, we're currently using a genetically modified yeast to produce a natural sweetener. In this process, we're trying to both optimize the cells themselves and optimize the biochemical process in which we use this yeast.
I'm the daily coordinator of the Erasmus Mundus program that is being coordinated by Leuven Campus Ghent. The program is entitled European Master of Science in Sustainable Food Systems Engineering, Technology and Business. The program is jointly offered by 4 European partners. Up to now, in the past 20 years history of the program, we have hosted over 500 students originated from all over the world. We have counted up to 75 nationalities that have visited us and participated in the program.
So far, more than 75 PhD candidates have successfully defended their PhD thesis within Biotech Plus. About 500 students have graduated from the International Erasmus Mundus Master Program Diponegoro Food Force. Towards the future, we keep investing in education and research, and we look forward to seeing our graduates taking up important positions in the domains of sustainable food systems and in the domain of climate change.
Do you know that your Professor Yoga is a graduate of that program? In 2012-13, he graduated from the previous edition of this program on our campus in Ghent. Right, microbes, negative microbiology, that means microbes can be a threat. They can harm our health, our safety, and so on. So let's have a little bit a look at negative microbiology and the research that we are conducting these days. I like it to be a bit interactive. So who knows what is a biofilm? Ever heard of a biofilm? The chairman of the session knows, so he can answer on your behalf. No. No, you?
Yeah, I think it's a film that's formed by the microbes, yeah.
Yes.
Basically.
Mm-hmm.
And also, anyone wants to answer? So, yeah.
No what?
Anyone?
No?
Biofilm.
You should know that the size of a microorganism is in the order of micrometer. Very, very small, not visible with the naked eye. That's why in most conditions they try to grow together and form a biofilm on this surface, for example, and that makes them stronger. They construct a matrix, EPS we call that matrix. They form that matrix, and by growing together they can better attach to a surface, and that is really a dangerous situation. These biofilms we see everywhere. We see them in the food processing industry, on the surfaces where we process the meat, where we cut the vegetables, and so on. The biofilm can be in the leads, in the pipes, in irrigation systems. They can be everywhere, and then when the microorganisms detach and they reach you, they make you sick. Now, that biofilm is a community just like humans. They're basically the most successful community on the planet. They exist longer than we ever existed and will exist, don't worry. We had a project some time ago on biofilms, also the mathematical modeling of biofilms, and understand how they act. Try to understand what makes them so strong. If we understand what makes them strong, we can try to destroy the bonds between individual microorganisms. And you know what is their secret? They talk. Microorganisms talk to each other. They use quorum sensing signals. They produce a certain chemical molecule, a chemical component, an autoinducer too, or whatever. The other organism is sensing this, is measuring this, and then they know we are too many, we need to move to another place, or we can still keep growing. Or it could be a biofilm consisting of different microorganisms. And then they are competing, who will win? Do you know that microorganisms even cheat each other? They pretend like, no, we are not that many. So they send out the wrong quorum sensing signal. The other one is not attacking them. In the meantime, they take over all the place. Sounds familiar, right? When I say they are acting like humans. There is a full domain, the social life of bacteria, that is developing extremely rapidly. Why? Because of the more detailed measurements that we can collect these days, we can better understand how they behave and how they manage to isolate or to, after some generations, one microorganism is winning the game. Game theory is at the basis of the understanding that we collect these days in biofilms. Now, biofilms are really a threat, and we should fight them. How we fight them these days is typically with chemicals. Chemicals have the major problem: you're polluting. So we're looking for alternative ways to destroy the bonds between the microorganisms. As an example, we have light. And in that project, we focus on light. It could be natural light, sunlight, or it could be monochromatic light. Light with one wavelength. In this case here, it was blue LEDs that we have been using. And in that project, we checked the effect of light on samples deposited on different consumer-relevant surfaces. Now, biofilms, as I said, the social life of bacteria, it's amazing. And you know what? They don't have mobile phones. So they still talk to each other. They don't send, and they send messages. That's then the other parallel. They keep sending messages, but they really have a mechanism of talking to each other, so the other microorganisms sense what is happening. These are real pictures. These are simulations that we do on the computer. In a biofilm, as in any microorganism, you have mainly two processes. They need to reproduce. They produce daughter cells. They multiply. This is the reproduction algorithm. DNA replication and cell division, right? And then there is motility. The one microorganism sitting here, it produces a daughter cell that needs to sit next to it or on top of it. But the maximum height is, of course, limited. They can only fall from the top. Modeling all of this, I know this is your favorite. These are diffusion processes. This models the motility, the movement of the individual cells in the biofilm, while at the same time you have the biological process of the organisms growing, pushing away other cells or pushing away cells belonging to another strain. And then you have competition between microorganisms in the same biofilm. There is a little movie here. I hope it works. There's no sound, no stress. So here, this is the surface. Here, and the cells grow horizontally on the surface, and then they move to the top. Now, the black screen initially is the available substrate. The substrate is, for instance, the carbon source they used to grow. When you see it becoming more light, it means the substrate is all used, and they have to look for another place where there is food for them. That makes them growing in different directions. I show this little movie again and have a look. They consume the nutrients and they grow in the layers. And the faster ones, they win at the expense of the slow guys. Recognize the picture? Yeah. And eventually win the race to the top.
Yeah.
These are real images. With SEM microscopy, we try to simulate this to understand better the mechanism. As I said, if we say we try to better understand the mechanism, there are a few things that we need to understand. This is cooperation. If you as a class want to survive, you need to cooperate, right? You need to help your fellow students. You all want, at the end of the day, in 4 years to obtain your degree. There is no limit on the degrees the university can offer, so you better help each other. You do practical work together, you write reports together, you study together, I hope. We're all social people. The organisms do the same. Of course, you have a few of these competitors. I want to be the first. I don't want this trait in the population. I want to be the first. I want to favor a certain It's not very sound as a principle, but this is what microorganisms do. To obtain all of this, they communicate. They send signals that are sensed by the other ones. What could be these signals? For instance, they could be gene transfer. It's not a mechanism. A very active domain these days, gene transfer leads to a modification in the genetic material of the original strain, so you end up with a population that is completely different from the original one. Could be a very dangerous situation. By the way, that is the reason why many of our antibiotics do not work anymore. The organisms have a self-defense mechanism. We treat them with an antibiotic. The survivors, they are more strong, and they are at the origin of an antibiotic-resistant strain. If you go to the hospital in Belgium, the first thing they check before they even want to consider what you have and what you need to be treated for, they check on multi-resistant Staphylococcus aureus. And that's a very dangerous one, because we have hardly any antibiotic that can kill it. I guess it's the same everywhere. We need to understand, these are more the mechanical stuff, adhesion forces. They attach to a surface, or they attach to a fabric. They attach to each other, of course, as well. We need to understand chemical gradients, oxygen, although most of these are in anaerobic conditions, but they need to have oxygen, some of them. Some of them need substrates. And if at a certain place the substrate is all gone, they move to another place. And they expand the biofilm. Metabolic networks at the basis of our understanding what happens at the level of one individual cell. Metabolic network describes the whole machinery of the cell. Of the microorganisms. You have heard of the Krebs cycle, you have heard of energy generation, ATP. This is all in the metabolic network. Horizontal gene transfer, I explained already how strains can modify, and signaling communication has been mentioned as well. Resource capture, well, they might even adapt to novel resources. These guys are so clever, you cannot imagine. They can survive in even the most harsh conditions, just like humans. We simply have to adapt to the new environment. That could take generations, but there was another thing there. That could take multiple generations, but at the end of the day, they will manage. If there's one lesson you should keep in mind, it's not to threaten you. Microorganisms are always several steps ahead of us. We can only try to remedy, to solve problems. But they're much more clever than all of us together. We should try to understand them better. Viruses are the worst. These are the parasites. They even use you as a host to multiply. Okay. Human societies, we try to maximize the speed of economic growth while exhausting our resources. Or we could opt for a slower growth but long-term sustainable growth. What do microorganisms do? A similar challenge. When they grow very fast, that leads to inefficient consumption of the resources available. Or if they sense we don't have much, solidarity comes into play. A slow growth rate is the result, but at a very high yield. They convert the last piece of food available, the carbon source. They're very efficient, and they obtain the maximum consumption of resources, so they generate the maximum number of cells. It goes a bit slower, but at the end of the day, they are good. Exactly the same dilemma as we have in human societies. Although we are focusing too much here, we also should focus here. What is the problem here? There are red cells and green cells, you can see that. So we have a biofilm. We start with a population of green cells. This is the surface. This is a cross-section. You see that it gets more white, so they're consuming the carbon source. Once it's exhausted, they grow more to the top. And what you see is that if the green cells are all next to each other and the red cells are all next to each other, they both survive. See that? It's only at this interface that you can see the reddish guys, they try to take over. But
So
they are not.
is it a different species or?
These are different species.
Different species.
Absolutely. The red ones try to take over, but the green ones, they grow more in block as a very solid community, and they can resist the attacks of the red ones who try to invade in random ways. Amazingly parallel, huh? Both can coexist. In this simulation, you see what happens. We did the simulation, a green cell next to a red cell. After a few cycles, the reds take completely over. Same mechanisms, same story, but the greens do not find their neighbors, they don't find their friends, they cannot defend themselves. It's one strain who wins the game. We see that in reality as well, also in the experimental data. This is what you can do with individual-based mathematical modeling, we call it. We will not go into the details. These are simulations. We try to validate this in the lab as well. Sure, we have a question I can see. Ah, no. Yes. No. Clear? Are you still with me?
Are you guys like how the microbes they compete each other as like a human, isn't it? Yeah, how they cooperate and make a social life.
Work together. Even within different traits, different microorganisms, we are all humans. Work together. The only thing that works.
Oh, sorry, and can we open up questions? There are questions from one of the students. Yeah.
These are two simulations, yes.
So one where species A is together and species B is separate, and the second simulation here where species A is embedded into species B.
Yes and no. And the previous one, very good question, thanks. So this is here, but can I have a better pointer? Ah, fantastic. Laser pointer. This is here the surface. This is the desk. And I have here, I inoculate with the green species. I inoculate next to it with the red species. And then I let them grow. And we see that the red ones, they try to invade, as you see here, take over, but they grow in such a compact way, the green ones, that they can resist the attack from both sides. If I now inoculate this surface, theoretically speaking, of course, with one green cell next to a red cell, but I cannot stop this little film, but this happens here, you see it here. We have a few green, a few red, a few green, a few red. The green cannot win the game. They're not strong enough, and they're overgrown literally by the red ones. So the spatial distribution, the initial spatial distribution is key in who wins the game. Yeah? Does that answer the question? Thanks. More questions? Oh my God, I have questions. Amazing. I'm so happy. Yeah, no, really?
Any questions? Yeah, for me, probably a simple question. What is the red one species and the green one?
This is simulation.
Ah.
But it could be an E. coli
Any.
together with the Listeria.
Any bacteria.
Any. This is the simulation. Yeah.
Mm-hmm. Okay.
In reality, there is no two species biofilm. Eh? There will be an immense number of different strains in the biofilm. When you isolate it, you will do selective plate counting so that you see, we have this one. You take another selective plate counting, we also have this one, or you do a full analysis and then you see all of the strains that are available or that are present. But it's the same that happens in our gut. I come to that much later, if we have the time. After a while, you out-select and the strong ones are winning.
Problem with the microphone.
This is not working?
Yeah, for the hybrid session.
Okay. This is better? Yeah? Okay. I was saying? Yes. After a while, you out-select and then there are a few, what we call dominant species, who win the game. But in essence, you start with a multitude of different microorganisms. Yeah? So far the biofilms, although not yet. We are also working on developing antibacterial nanomaterials. What is this? If I have a surface and I want to disinfect it, I could try to use light, or I could use a chemical, or I could use heating. It's not always possible, not very clever. Light is definitely an option. Nanoparticles is another option. I could even try to make my surface resistant to microorganisms trying to attach to that surface. And that means I need to develop what we call a coating that has antimicrobial properties. Do you know that even in the textile industry, this is a very hot topic, that we are developing clothes, fabrics, that are resistant to microorganisms. These things exist already today. Also in the fabric that we use in cars, Fiat, for instance, the company Stellantis, they are partner in the project we are doing. They're very interested in developing this kind of fabrics to make them resistant against microorganisms. In a rental car, many different people, many different continents. If the fabric, the seats and so ever, the dashboard is pushing away the microorganisms, there is no problem at all. If you can make the surface with a coating, consider it like a paint containing a few nanoparticles, you can do a lot to prevent contamination, to prevent infection. And this is what we do, of course. You need then to develop these antibacterial materials, these antibacterial nanoparticles. We synthesize them with novel technologies like plasma. You know, plasma is the fourth state of matter. We have solid, liquid, and gas, and plasma. Plasma is that very weird state that we try to avoid to describe. We don't see it in primary school. They mention it the first time maybe in high school, but at uni they mention it. It is that ionized state of matter. It is in between all the other three. But it is a very specific one, and it helps us to synthesize these extremely small nanoparticles. We use different anodes, we use a different cathode, and with some electrodes, we can generate the plasma so that we induce or that we form these nanoparticles in this liquid solution, for example. These metallic nanoparticle coatings, silver is an important one, zinc is a very important one, tungsten is a very important one. We can use these particles because we can then test in the lab what is their antimicrobial power, what is the antimicrobial property of these nanoparticles. We do a very, very thorough screening and we try to synthesize the nanoparticles that we like. Which one do we like? Those that are fighting the microorganisms the best. And this we measure in number of log scale inactivation of the microorganisms. There is treatment with antibiotics. We have antibiotic resistant strains. Solution, forget about this. We cannot keep inventing in the pharma industry novel antibiotics, let's try to fight them in another way. Coatings. As I said, we do in the lab a lot of in vitro antibacterial assessments. Once we have synthesized a new nanoparticle, we can test it against a single strain. That is then nice, but that I would say is more an academic funny game that you can write a nice paper on and a nice report. It's not reflecting reality. You should fight a cocktail of bacteria. This is what happens in reality. And then keep in mind, in that cocktail, some of these bacteria are working together to survive. So you should design your nanoparticles in such a way that you destroy the strongest ones, or at least inhibit the further growth. These are two different things. So that your surface stays safe. Once we have done this in a liquid, we try to do it in a kind of a polymer. We try to do it, our nanoparticles are immobilized in this kind of coating, typically on the basis of a polymer. That coating we put on the surface, and we have to do the tests again. This is what we call the in situ bacterial assessment. And this could be the surface in a car, this could be a fabric, it could be the surface in the food processing industry, an inox surface, and so on. You will, or you might be surprised, even if it looks extremely flat, if you look under the microscope, the surface is extremely irregular. And it's in these pit holes that your microorganisms found shelter, and they start growing as happily and readily, and when it's too late, you see it. This is what we do in a multinational project in Europe. It's called the SUPREME project, which stands for Sustainable Nanoparticles-Enabled Coatings with Antimicrobial Properties. We have about 15 partners in that project. We are a little bit more than halfway. The in vitro, that means in the liquid, in test tubes in the lab, tests are now finished. And we now have identified the most powerful nanoparticles. And we now use these particles to make the coating on a fabric, on a metal surface, and test again. And then see what is the most efficient one. In the mathematical modeling that we do, we try to understand why it works or why it does not work. I'm not going into these details here. But if we would have, you can do, let me start over. In the pharma industry, if we design a new drug, the approach until the 60s, 70s was, well, let's try. Was a lot of trial and error. This molecule works, fine. But this one, if we modify it a little bit, this works better. We didn't have a decent strategy to design a molecule that works. Same story with the antimicrobials, with the coatings, with the nanoparticles. What works? You need a design strategy, and this becomes then more mathematical, more optimization based on the chemical properties of individual materials. And what we see now also in pharma and drug design, the initial stages are all in silico computation based. We outselect the most efficient, or we try to predict the most efficient combinations, and then we test them in practice. That might shorten the cycle of obtaining novel materials. Now this here was also a bit trial and error, but in parallel, we work on developing mathematical models that tell us why is that happening. Similar as the very simple example I have been showing in the beginning on the biofilms. We see these nanoparticles interfering with all these different strains. Why do they work? It's your job as academics not only to solve a problem, but to understand why it works. The main reason is there is conservation of energy. You know that. you're all perfectly trained in that in your courses on physics and so on. There's conservation of mass, conservation of energy. There is also what we call conservation of misery. You know that? You say yes, but I don't believe you. Conservation of misery means once you solve a problem, most often you have created a new problem. And that was the big drama with Alexander Fleming, with his penicillin. Long, long time ago. You could all be my children, right? You're so young. So I remember these days. Fleming invented by accident, he saw that his petri dish was overgrown by penicillin and there was no further growth of any microorganisms, so that was the first antibiotic, the penicillin category of antibiotics. The problem is, after a while, they stop working. And then you need to find novel techniques, novel technologies, you cannot keep going. You need a strategy, you need a mechanism, you need a design path to develop new antimicrobial stuff, drugs. Same story here with the nanoparticles. You can try to do it, trial and error. It's better to test them in silico, develop, try in vitro, that is high throughput, and then test on the real surface. But that's a very tedious job. We are at the beginning also because we can collect much more information than we could collect in the good old days. At level of the microorganisms, we can basically collect all information that we want. Omics is there. Okay, so in this project, the Commission launched that call, and we could obtain one of the 7 funded projects. And they wanted us to have nanoparticle-filled coatings with recognized effectiveness against bacteria, fungi, and viruses. This was in the aftermath of the COVID. And the European Commission said, we need to protect ourselves better. We want you guys in the academia and in the industry develop new coatings. That was at the origin of what was happening here. Okay. In the lab, we also work on novel decontamination technologies. Decontamination, it means we inactivate microorganisms, we try to kill them, but if we don't kill them all, they might end up in a dormant state. They could be reactivated later on. Be very careful what you do. There are 2 things. There is inhibition. Inhibition is nice, but that simply says they stop growing. But they're still there. What you want is kill them. To kill them, we need to better understand mechanisms. What kills a bacteria? Can you help me? That was a question. Hello, question? People online, question? What kills a bacteria? Salt? S-A?
O-L-D.
A soap.
Yeah, soap.
Soap. Amazing. Yeah. No. Soap, yeah. But, yeah, I see your answer. And then what happens to the bacteria? I don't know. Why is it killed? Why is it dead? I like the soap. Do you know how a bacteria looks like? No? If you didn't see that yet. There are gram-positive, gram-negative bacteria. The main difference is at the level of their membrane structure, the envelope around it. Let's not make it too technical because most of you have not seen this. But see it as a big bulb, an envelope containing all genetic material, mitochondria, the whole stuff is there. If you destroy the membrane, it's finished.
Mhm.
With pulsed electric fields, we try to penetrate the membrane at such a level that it is destroyed. That is a novel technology being used in foods. The Japanese, they have already fruit juices pasteurized via PEF, pulsed electric fields. You could try to crash them with high pressure.
HPP, isn't it? HHP, high hydrostatic pressure.
High hydrostatic pressure, very effective, being used. What do we see? That's a funny story again. One of my colleagues in Leuven, he created a high pressure resistant strain. In the following way, you have an initial population. It undergoes high pressure, thousands of bar. There is one survivor, that's enough. Let that survivor grow. Subject it again to that high pressure. If there's one survivor and you keep repeating that cycle, you end up with one strain that is resistant to the high pressure. So whatever we do, they always find a way to build up resistance. That final strain was completely different than the original one that you tried to kill. That is true. But then at the same time you have played a bit Frankenstein, you have created another one that is now resistant to high pressure. And that is now out in the environment. But it still is pathogenic. That is the main problem. Other technologies are radio frequency. Radio frequency, especially for food, has the nice property that it penetrates into the food and that it leads to uniform heating. More uniform than traditional pasteurization technology based on the heat. Okay? Radio frequency, it penetrates, that depends on the wavelengths, it's in the order of megahertz frequencies, and they penetrate in a more homogeneous way in the food so that you have a more homogeneous inactivation of what's happening, and you're not overcooking the boundary of your product while you're killing what you tried to kill. We use that for enzyme and microbial inactivation. You see here a radio frequency setup built by one of my postdocs in the lab. As you can see, he builds everything out of waste. This is all electronic waste. Of course, the oscilloscope and the generator we had to buy, but the real setup he created himself. Another technology is non-thermal plasma. We have been mentioning this. I illustrated already the plasma at the level of using it to generate nanoparticles. Okay? Here we use the plasma because the gas, when we start the plasma, we have ionization and of course in equilibrium with deionization between the gas phase and the plasma phase. We have a lot of what we call the most active species or the reactive species. And these reactive species, these ions, these electrons, photons, you name it, these play the role of the antimicrobial agents. And they try to kill the microorganisms. So you generate this plasma. When you put the microorganisms there, for instance, this is a plasma, a dielectric barrier discharge setup that you can use for surface treatment. If you have, this is the surface, this is the plasma generating device, we have the discharge, and the plasma reaches the surface and it kills the bacteria. Not the plasma itself, it is the reactive species that kills the bacteria. Designing this is what we do in the lab as well. And what we could see after more than 30 minutes, we have an inactivation of 1 log. That was not much, but you should keep in mind we are fighting biofilms here. Another setup, what we do now in low moisture foods, these are typically seeds, grains, all of that stuff. This you cannot simply pasteurize because it's not liquid. There we need other technologies where, for instance, we create a fluidized bed. I don't know whether you have seen it in the movie, but we have a tube where, with the air coming in here, we constantly keep the particles in movement. And then we can have our technology, whatever technology you want to use, so that we reach every side of the particles. That is crucial, because if you try to treat a milk powder, very interesting stuff for babies, by the way, milk powder, the contaminations, there are quite a few cases reported. So you need to treat the individual particles in the powder in such a way that you reach every side of the surface, that you keep them in movement, that it's not only the surface, because then it will become brown and you're burning your powder from the outside. You need to reach the powder completely. That's why we built this fluidized bed, so that we can reach the full particle surface. That's the topic of an ongoing PhD study as well. At this moment, as a scale not bigger than this, the fluidized bed that you see here. Now, what do we want to get out of these studies? That is valid for radio frequency, it's valid for non-thermal plasma, it's valid for many, many different applications. We developed that fluidized bed, but we try to understand why it kills. The simple observation that it kills, that's high school level. That's fine. But it does not help you to make progress. As academics, we try to make progress because we understand why something works. If we understand why it works, we might even improve our technology. Make it more specific, make it more selective. And that, for instance, is what we try to see here. We mentioned already as a possible mechanism that we damage the membrane. It could also be that we are damaging the double-strand DNA. There are very specific techniques that allow you to measure what is membrane damage, what is dsDNA, the double-strand DNA damage. We can measure this. And on the basis of this, my PhD student Julian is now working on, it's for an E. coli organism, what exactly is at the origin of the inactivation of this organism. Is it membrane damage or is it DNA damage? Once we know this, we have a novel antimicrobial therapy remedy that could kill similar microorganisms, bacteria as well. This is a bit of an overview of negative microbiology, where we try to kill microorganisms. Let's now move a bit to positive microbiology, where we try to exploit microorganisms to the benefit. They do something useful for us. But I need a small break, and you might be needing a small one as well. Shall we break for, I'm looking at Mr. Chairman here.
Yeah.
What time is it now?
It's 10:30, I guess. Yeah.
10:30.
10:30.
No, that's
Shall we
not true,
continue?
eh?
Shall we continue, or do you need a break?
5 minutes.
5
max.
minutes. Okay.
If you want to stretch your legs, do it.
Yeah. If you want to go to the toilet or
5 minutes, not more. Is it really 10:30?
10:30, is it?
Are you still with me? Are you still happy? I'm not looking. Good. Allez, 4 minutes and a half.
4 minutes and a half. Yeah. If you want to go to the toilet, please help yourself.
I need to stop at 11, right?
Exactly.
Yeah, sure.
We still have time.
You know it better. These are first class students. Is it too fast or is it too slow?
Well, there are some material they could understand. I mean, most of them, when they are trying to apply UNDIP, they are from very good. Like for instance, when they are applying for studying here in UNDIP, they are already a very bright students. Most of them don't understand.
No. All right.
It was very interesting, especially about how do you eliminate some novel like radio frequency. It's something new.
I didn't.
Yeah.
Well, we use it already for thawing. For thawing, defrosting material. There it is used in the food industry, but to really inactivate it is new. Yeah. It's not the most effective one, but at least it does not destroy the other properties of the food. Organoleptic properties, the structure, the texture, which is the case in many other fungal technologies.
When I was a master, my thesis was about EAF, the pulse electron field.
Yeah. You did it too?
No, in Hungary.
In Hungary?
In Budapest, yeah.
With who?
Do you know anyone from Hungary?
I know the team of Laszlo Meseros and Joseph Farkas and Silvia Farkas and these people. Do you know
Yeah, they're from another laboratory.
Okay.
Laszlo Abramko.
Who?
Laszlo Abramko.
Yeah, I know the name.
Yeah, I did working with this analysis. Yeah.
Is it UHFI University? Or what is the
Martin. It was a Sam name, but now they changing
Okay.
the name to Martin.
Okay.
Yeah.
Interesting. Ah, you were in Hungary.
I was there, yeah.
As an exchange student.
Pardon?
As an exchange student, or you
No,
did the full master?
Full master there.
You speak the language?
Not really. It was a very hard language.
I know. That's very difficult. I should switch this off or don't touch?
Yeah.
Don't touch.
There's a question student wants to ask you questions. Would you like now or?
Shall we start? Okay. Okay, so attention everyone, so we're going to start again. But there's gonna be, I think, a question from some students. Anyone would like to ask a questions? Okay, please. Okay.
You want to use the microphone that the people online can hear the question as well?
Thank you. Okay, hi, Professor. I hope you still remember me. I have a question because when we talk about the negative of microbiome and we connect it with the food, I'm pretty sure most of the people here will agree with me that one thing that comes in our mind is free meal, the program for our Indonesian presidents that cause food poisoning for the students. Approximately thousand of students get a free food poisoning because of that free meal program. And then what makes me wonder is that they use the trays for the free meal. It's reusable, I don't know until when, which is increasing the risk of microbial community live there. So my question is, do you have any strategies to kill or at least eliminate the microbial community that live on the trays without without making them resistant? And is it possible to do it on the massive scale, like for the whole nations? Thank you.
Very interesting question. How can I reply? I cannot go into the amazing plan of the president, or who did he say? That he's offering free meals, that is fantastic. I don't see directly the relation between the free meals and the trays that get contaminated. But okay, there is something going on. Do we know for sure that the main source of contamination is in the trays?
Yeah, there are some news that talk about the possibly improper cleaning of the tray. Yeah.
It is not at the level of the food that you eat itself. It's a question, I don't know. Could people identify that the contamination or that you get sick because there are microorganisms left over on the trays? Or is it a contamination because it is a free meal? All of the students like to eat that meal, so you have a mass production of food, and maybe the food was not heated sufficiently for all the students, I mean for that massive quantity. Do you have already investigated what is the real root cause of the problem?
I'm pretty sure it hasn't identified yet whether it's from the food or the tray, because those two have the possibility.
At first sight, these are the two options, yes. Yeah, but this is not cleared out. What is the reason, or is it?
This is like a complex situation. I mean,
Yeah.
like they could happen because of the cold chains, also the transportation and etc. But what she was asking, the strategy, like the massive tray, how do you properly clean it? So there is no leftover, isn't it?
Do you know how the trays are, when you say they are not cleaned, do you mean they are used from one student to the other, even if it's a bit dirty?
They reuse it.
Yeah.
Like they give it to the students and then they will take it and then they will give it back.
To another student.
The one who clean it is like staff of this catering.
Yeah. So they collect the trays and they don't clean them when
Unproper clean.
a simple cleaning in hot water is more than enough. It's already more than enough. And if you, but I'm puzzled when you say that. Did you say 1,000? Did you say 1,000 of infections?
The number of infections. It was 5,000 cases, like
5,000 cases.
in whole national, like 5,000 cases of.
Yeah. That is a very serious problem, but I do have my doubts that it is via the trace. I mean, they don't have the time, the organisms do not have the time to follow a resistant biofilm that could be very harmful, because it's always in circulation, it's always being used and reused by other students and so on. I would more go into the preparation and the full chain of the preparation of the meal. It's a very interesting question. It's a very interesting problem. Unfortunately, students died. That should never happen. It has to do also with the fact, and we did a study in Leuven as well, do we prepare the food locally on the different mensas, restaurants, whatever you call them, on the campus, or do we have a central kitchen, and do we have a system to transport the food in a safe way?
They do have the
And
central
that-
kitchen, yeah. So for instance, like one central kitchen, they cook in the morning and after they serve in the afternoon, like 6 or 7 hours of preparation. Yeah, they could also have
There is your
problems.
problem.
Yeah, they could
If
also be
the
a problem
temperature
as well.
control is not rigorous enough, you could have a problem. 7 hours is enough. Microorganisms, they start growing at ambient temperature like crazy. If they, and they are in a food, so they have lots of food themselves. In Leuven, we developed a sous vide system where the food is packaged under vacuum, even the food is ready. This is then transported to the different local restaurants and then reheated and served. But if you say that the food is prepared in the morning and could be staying there for a long time, and then maybe slowly or just a little bit reheated, or if not, then you could run into problems. Did they identify the specific bacteria that was responsible? No.
It could be Bacillus or Salmonella. Yeah.
Yeah. I want to discuss this at some point further. Yeah. Wow. Very tough problem. I mean, you try to do something good, but yeah, the food chain should be monitored very accurately. That is the main thing. And it is typically in the last step, also for all of us, in the last step, the reheating of the food, we are a bit sloppy ourselves. Now, I'm not saying in your specific example that was the case, but if we eat a pizza that we collect at the street, temperature might be way too low and it could have been lying there for hours in the sun or in the heat. And in the humidity that you have here, that's asking for problems. And that's why in your kitchen you have so much fermented food and spicy food. That helps as a protective effect. Cleaning it with soap, that could be an option as well, but and even just a bit of hot water is enough for the tray. But you're young people, I have problems to imagine that you could not resist what is happening on that tray. You're not eating from the tray. There could be a bit of contamination, yes, but you're young, strong people. I think it is more food-based than tray-related. That's my first guess.
Okay. Any questions? Okay, please.
Oh, okay. Thank you so much. So, can you please help me to show the slide that shown the high frequency radio technology, please?
You want to see that slide again, you said?
Yes.
Google Translate is amazing, eh? Congrats that you dare to ask a question. Let that be clear. This one?
Yes, thank
Yes.
you so much. So, this technology is very interesting for me. As a food technology student, I'm pretty sure that nutrient content is a big concern for us. So, novel technologies using high radio frequency. We know that high frequency can create heat in materials by the electric heating process. So, how this technology ensure that there is no damage for the kind of nutrient that sensitive into thermal process? So, what is your strategy to keep the nutrient safe and there is no damage during this process? Thank you so much.
Very good question. Excellent question. There are a few answers to this. I cannot personally guarantee anything. Answer number one. Secondly, we always talk in comparison with traditional thermal heating. You cannot avoid that the radio frequency will not heat up a little bit the food, but it will definitely be at a lesser extent. And not at an extent that it kills the microorganisms. It helps in combination with the radio frequency, the waves themselves, and not at the extent that the food, the texture, the organoleptic properties, the rheology, the vitamins are killed completely. But you're fully correct, we need to investigate this in full detail. At least it is what we consider, with respect to these properties, a mild technology. But you cannot, I do not dare to claim it's non-thermal. But it is less temperature intensive. The second thing that is interesting, and now we go into physics, it is a wave. Now it is well known that with waves you can heat up a product with a less increase in the temperature than with just pushing a thermal heating at the surface and then wait until this propagates. Waves have the property of propagating better inside the food at the expense of a less increase in the temperature. Since this is also a wave-based technology, that helps. But perfect it is not. It is a technology that comes from a thawing. Thawing is the defrosting, the heating up a little bit of material where it is quite used. And as you can understand, it's from typically Scandinavian countries use it a lot. Deep frozen fish, when you defrost it, should still be okay. We don't want that you also have a lot of frozen fish here, I imagine, seafood and so on. We still want it to be a little bit crispy and not something very less tasty, let's say. So there the technology is proven. Of course, that is not intense enough to kill bacteria. We need to increase the power that we transfer via the technology so that we have less bacteria, and that is then a trade-off problem. Organoleptic properties, the quality of the food, vitamin content, other properties, versus the taste even, and the biological safety. That trade-off you need to make in any case. But it is in any case a temperature increase less than with traditional heating. That's for sure.
Yeah, thank you.
Okay, shall we continue the next slide? Or
I thought he had a question, but you're
do
the
we have any
cameraman.
questions?
Yes, please.
Okay.
Okay, thank you, Prof. I want to ask, from your topic, I noticed the discussion about technologies to address the negative side of microbiology to food, and especially related to microbial contamination and biofilms. I have read about antimicrobial edible packaging and with controlled release system, and I'm curious about the role of antibacterial nanomaterials in this context. How do you see their future in food microbiology, both in terms of effectiveness to kill pathogens or for safety for us? Thank you.
To kill?
To kill pathogens or microbia.
Thanks for the question. Very interesting question as well. You will answer it yourself. If you go fishing, suppose you go fishing, we are very close to the sea. What will you find? Hopefully a fish here and there, snapper, what is a seafood, but what else will you find? I'm pretty sure. When you analyze a sample from the sea, what will you find?
Maybe some microbiology, like bacteria or...
Also? Okay, these are the nice guys. There's some plankton, there is some small fish, big fish. What else will you find? And that's rather recent.
I don't know.
Yes, you know.
Oh, plastic. Like
Microplastic. You might even find it in the fish.
Okay.
Do we like that?
No.
Is it then thinkable that we, what you said, the eatable packages,
Yes.
packaging method and so on, that has antimicrobial properties? Can we use nanoparticles for that?
Yes.
In direct contact with the food, that is a tricky question. By which I mean, is it safe for humans to have too much nanoparticles inside us? And that is the real problem, because these could be airbound as well. The thing is that in the project I was mentioning, that's from the Wageningen University, the one in the Netherlands, is their major university in the bioengineering field. We have a team on toxicology. And they are doing in parallel the studies, how much of these nanoparticles that we develop can we, human speaking wise, tolerate. But my advice is we should not. I would say we should try to avoid them to be in direct contact with the food as much as possible. If you can guarantee that they stay immobilized in the coating, I'm happy. But I don't want them to penetrate inside the food. That would be my personal advice. The eatable packaging, I'm not going to eat nanoparticles, me as a person. Although people might claim, yes, but the quantity is so little and so on and so on. Meh. I would not trust it in the long term. It's exactly the same what we see with microplastics. We even now see it in muscles of animals. And that's amazing. Everywhere we look, we find not nano, but microplastics. Do we like that in our food? Again, same question. People will say we don't eat that much, and at the end of the day, most of it is not leaving our body anymore. It's the same when I started teaching. This did not happen to Dr. Yoga, I'm pretty sure, but when I started teaching, I had to pass an X-ray. Can you imagine? Because these days it was not in the 60s, I'm not that old, but still, that was a leftover of the regulations by law. Tuberculosis is a problem, you cannot teach X-ray. The thing with all of these technologies, nanoparticles, is they don't leave your body, they slowly build up. And at some point you reach a level that it gets problematic. And that's, to my opinion, what I would not prefer to see happening with nanoparticles food related. They have a lot of potential. The antimicrobial efficiency can be amazing, especially the copper ones based or the zinc one based. But to have them in eatable packages, I prefer your fantastic food over an eatable nanoparticle based packaging. We don't digest them, they're inert stuff.
Thank you, Prof.
My pleasure.
Thank you very much. Shall we continue to your slide? Yeah. I think we can continue to your presentations.
But it's 11, eh?
Yoga, 11:20? Is it 11:20? We still have 20 minutes.
You want me to rush, eh? You don't let me go, eh? Ali, ask more questions. I only have this slide, there is nothing left. No, it's not true. Here we are, food sim. simulation of food digestion. It's our new toy in the lab, where we try to mimic what happens in our intestine. This is the human digestive system. Maybe we have a few Indians here. I don't want to offend anyone, but we have only one stomach. The cow has multiple. Still, you could do exactly the same analysis for any living animal. What we consider is not what happens here. I will rush a little bit. We start from the stomach and all what happens here until the exit. Yeah? So there's gastrointestinal tract. All of these solid organs there play a role, but we follow the food after it reaches the stomach, and so on and so on. Small intestine, large intestine, exit. There are three key players that we consider, and this is a very hot topic. How can we keep the gut microbiome? All of these guys that are not your enemies but your friends, they keep you happy. How can we keep them happy? So we have the gut microbiota. We could get sick, and then we typically go to the doctor and we take antibiotics. Now, before we can do all of this, we first should understand what is happening here. So there's a consortium of gut bacteria. We do experiments with two pathogens, gram-positive, gram-negative, and we work with two broad-spectrum antibiotics, ciprofloxacin and tetracycline, which is a family of the penicillins. Now, this is our model. It's not a mathematical model, it's an analog model of what happens in our system.
Mimic.
The food comes in this reactor. It goes to the upper small intestine. It goes to the lower small intestine. And it goes to the colon. And then our guy needs to visit the toilet twice a day. We feed him or her twice a day as well with a food system, a food model system that has the same essential ingredients as we eat. A Western diet, not your diet. And we follow what is happening in this system, which is a continuous system, although it's intermittently. The volumes are proportional, relatively proportional to humans. That means the stomach volume is half the volume of the upper small intestine, the lower small, and so on and so on. The colon has a big volume. And that is how we designed the different reactors. To model absorption processes exchange at the level of the intestines, we have this absorption process that you see here with this membrane technology that we took from dialysis. Patients who need dialysis, we use these membranes there. So with this system, we mimic what happens in the human gut. What is the purpose of this? We could do many, many different things. First of all, is the community of microorganisms, is it stable? As we have seen in the studies on the biofilm, there is a multitude of microorganisms present. Are they stable? Related to what we eat. There's a big interest in prebiotics and probiotics. Are these really helping us, or is it just a commercial bullshit to gain money? By the big companies, Yakult, Kraft, and Danone has its Actimel and that kind of stuff, right? Is this real, or is it not real? Why? In the Mediterranean countries, people have hardly any gastrointestinal problem. In the northern countries of Europe, many, many problems. Why? In your country here, the gut microbiome is completely different than mine. What keeps you healthy? To try to understand this, you're more in a position to see how you can defend yourself when you're attacked. When you're attacked by a pathogen, can you count on your self-defense mechanism of that big community of strains in the gut, similar to the biofilm, but now in a positive sense, Or do you really need antibiotics? Or could another component less harmful be helping? The main problem with the antibiotics is that it kills all the good microorganisms as well. Not only the pathogen, but also the good microbiota. And then you need to start up again to build it up. Could we avoid that? So at the level of human health, drug design, we believe this could be a very nice system where we do not need humans to do experiments with. That can come in a later stage. So that simulator, you see it here in the lab. It's fully computer controlled. And we have here, we measure everything. We do measurements of the weights so that we can see, okay, we have the food. It goes to the small intestine, the ileum. We have the absorption process. It goes to the lower small intestine in an intermittent way or in a continuous way, just as it happens in our body. Then it goes to the colon, and then we have the effluent, to say it in a clean way. Okay? And this is the system at real scale as we have it running in the lab. First thing that you need to do is make sure that the hydrodynamics, the flows, are correct. So we have to program, in a computer-controlled environment, we have to program the flow of eating twice a day, intermittently feeding the stomach, just as it happens here. That valve is not open all the time. The pH is extremely low in your stomach. You know how much? Could easily be 4 or 5, eh? Which is acid. Eh? If you have a reflux, you know. Eh?
Yeah.
Yeah. I guess with you it's even 2, with all your spices. I'm joking. I'm joking. Hello. Yeah. Now I'm lost. Yes. 4 in the stomach. So that valve is only opening intermittently, while here we have kind of a plug flow reactor system where we push slowly. There is time for the absorption process and so on. Of course, we don't sit on the toilet all day unless you want to check your phone. No, we don't do this. So this also is intermittent. Also, this needs to be programmed in the system so that we can really mimic all the processes that are happening. This is the cycle of one feeding cycle. We have 2 of these per day. Okay? I will not go into the details here because we are already over time, but I hope you see the possible large potential of a system like this where we mimic a bit the chemical engineering approach, and we don't need to bother you and feed you with stuff that you don't like and do. Yeah. The main advantage is we can also monitor the microbiology in the different stages of the process. And we can monitor what's happening with the gut microbiota here, what is happening here, what is happening here. And how is it influenced by the food? Once we get sick, what is now the best strategy? To get healthy, that is one objective, but to restore the gut microbiota as soon as possible. Or maybe find a strategy that enforces the defense mechanism of your gut. Then we don't need antibiotics. That's the domain of the prebiotics and the probiotics. And they claim we can do a bit better here. That is one example of positive microbiology. I suggest this will be my last one. This is in the domain of, no, I have one left, extra. Last but one. Bioproduction of tomatin. Who knows what is tomatin? I know that you like sweet stuff. Yeah, eh? Last night I had cocoa water with that syrup. What is the name again?
Which one? The fruit.
No, it was a liquid with ice, cocoa, and then that syrup.
I think it's ronde, you know. Ronde. Like something sweet with something.
Yeah.
That's it.
On the plane they tried to sell me sweet pancakes with vanilla. Yeah. Fine. Tomatin is a sweetener. It has a sweetness of a few thousand times glucose. The thing is, it does not make you fat. Fantastic. But we find the tomatin only in the catemfe fruit, which is in Latin America. And the concentration is not high enough, so we are looking for a way to produce it via a genetically modified organism, Pichia pastoris. That one has now a new name, but you might remember it from that name. So we try to produce it at a high level as we can find in the catemfe fruit via this modified microorganism, where we control substrate, temperature, pH, the media, and the supplements in the medium. I'm not going into the details, but we obtained results that are quite nice. And this is when later on, your fresh students, later on in your studies, when you see a little bit about fermentation technology, bioreactors, the two competing objectives here are the yield and the productivity. That's important for your bosses when you go into business. Productivity is what you produce per unit of time. Yield is the efficiency. How much of the substrate that you feed to the process is actually converted. And these two are conflicting. If you want a higher productivity, production per unit of time, you do not necessarily convert all of the substrate into the final product of interest. You lose raw material. In some processes, especially in the medical field, the raw material is so expensive that you cannot afford it. At the same time, to get the last bit out of the raw material, then you're focusing on yield. That might take an awful long time. So you see the conflict, productivity, time is money, and using the raw material. There is a balance, and you can see here a sketch of yield versus productivity. The one is optimal at this feed rate. The productivity is optimal at this feed rate of the substrate to the reactor. And you see that's a conflict. We need, or you need to talk to the boss and say, can you live with this or this? What do you want? The price of the product that you produce versus the consumption of the raw material. That's an economic problem next to maybe an environmental problem, because if you lose substrate, what will you do? Or is it possible to recover it from the stream, and so on and so on. One example of positive microbiology where we use a genetically modified strain to produce something of interest. A sweetener, low calorie. Problem with this guy, I call it the guy, because he has bad qualities. The size of that molecule is very small, of the tomatin. The sweetening effect, don't get mad with me, no. The sweetening effect is extremely high. What is the immediate consequence? I am happy. I'm interested in the sweetening capacity, but the people making the cookies are not happy. Glucose and its associated sweeteners, these are big molecules. They also have a structuring effect in your cookie. If I take out all of the glucose and I replace by my tomatin, it does not look like a cookie. So I need then to find a substitute for the structuring effect of my cookie to have the same sweetness, and still that it's a crispy cookie. Next to that, there are other sweeteners that we don't like at all, right? Can you name me a few? Other alternative sweeteners?
Stevia.
Stevia?
Yeah. Aspartame.
Ooh! Also, yeah. You like these? Which one do you prefer? The real stuff, eh? If you want my advice, if you sin, do it once per week. Don't drink a small glass of Coca-Cola, the regular, not Cola Zero. Drink 1 glass of Coca-Cola, even a full bottle, only once per week. Don't drink a small sip of Coca-Cola every day. That is bad. Sinning once and then get clean, no problem. Your body can handle. But having, I have too much sugar, I should eat less. No, don't. And sin once a week. You will be healthy. If you remember this from class, I reached my mission. I will not go into this, just switching to the last one, but we do a lot on mathematical modeling as well. I know you hate it, so that is for tomorrow for the conference. But I give you this example here to conclude. Alpha fuels. This is designing new fuels for aviation industry. Non-fossil fuels, a very hot research domain as well. Sustainable jet fuels from CO2 using microalgae cell factories in a zero waste approach. Wow, 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 that sounds impressive. Whew. It solves all the problems again. Yeah. So we capture the CO2, we feed that to a genetically modified microorganism, and that modified microorganism is not producing kerosene, of course, for the plane. It is producing a precursor for a new aviation fuel. It's producing isoprene. And isoprene is a precursor of many modern fuels that we can then mix. It will not fly yet at 100% of that new fuel, that we can start mixing with the traditional and then reduce the importance of fossil fuel. You don't want to know before the plane is taking off how many tons of fuel you have already used. If you're flying a big aircraft. Yeah. So the impact there is really big. Now this is a bit of commercial talk. What we do here is basically the following. Cyanobacteria, for instance Synechocystis, pardon. Some people classify them as algae. Okay, let's not go in that eternal fight between microbiologists. Cyanobacteria, they need CO2, so we solve the CO2 problem a little bit. They're also photodynamic, they need light. And that induces the machinery, since it is genetically modified, they will not only grow, but they will start producing isoprene. And that isoprene is a very essential building block in the chemical industry. Polymerization leads to rubber, synthetic rubber. Dimerization leads to sustainable fuels for aircraft, for ferry boats, and so on. And that is the objective of this project here. We are now in my lab at the stage that we go from CO2 plus light into production of isoprene. What are our degrees of freedom? Well, we need a certain level of the substrate. We play with the light. It could be day and night cycles. If we want to do that in an open pond in nature, then we could use or make use of the sunlight to do that. Or do we need to go to monochromatic light? Is the efficiency, the yield then better? Then we have to do it in the lab, but then we have a problem with scaling up our process later on, because we really need massive production of the isoprene. These are the degrees of freedom in this specific process. We now do it at lab scale. Our job is to come from design an optimal cycle of the light that we offer to the cyanobacteria so that they produce the isoprene in an as much efficient way as possible. What was the problem with the young lady with her question on radio frequency and it's not heating up or it is heating a little bit? It had to do with the penetration of the technology. The heating, when we have a wave, it penetrates better. The light is exactly the same problem, especially at scale up. If you have that massive big cylindrical bioreactor, it's not going to work. You need then maybe a ring or a setup, a Christmas. Yeah, you don't have Christmas. You celebrate Christmas? No, huh? Anyway, Diwali? No, that's for the Indians. Whatever, Feast of Light. You have that ring of light that you can put in the reactor, or we work with a flat plate reactor. And that's the setup that we are using now. But for this you need a lot of surface, yes and no. You can put all these flat plates next to each other. And this is how we see the upscaling of that process. The upscaling is not our job. We have the small flat plate in the lab. It's about the size of an A4 of your laptop, that we have a bit of a size. It are the colleagues in RISE, the Research Institutes of Sweden, who will do the scale-up of that process. We are now about halfway this application. These are a few examples of positive microbiology, what we do in the lab. For food, for feed, for pharma. You have a lot of fermented foods. You can experience it every day. One final word. Don't be too clean. Take care of your immune system. Don't start wiping it clean with Dettol. You know Dettol, right? Every single day, you're simply destroying your own immune system. I'm not saying that you should eat bacteria, but keep a balance.
Keep balance.
And after all, they are our best friends. They trigger our defense mechanisms in such a way that we stay strong and healthy. And that's what you will need to finish this degree in about 3 years and a half. I wish you the best of luck. It has been a pleasure and an honor to teach to you. Teach. Tell some stories. Any questions? Be my guest. Thanks a lot.
Okay, give them applause, please, everyone. So moving on to next questions. Anyone would like to ask questions? Yeah, okay. So these are our students from Malaysia, is it? Yeah.
Tell me your name is Najwa.
No.
I had a PhD student from Malaysia.
My name is Mai. I'm from International Islamic University Malaysia, here under the Diponegoro Exchange Experience Program. So my question is, you mentioned before the first one is treatment with antibiotics. One of the example is fabric resistance toward microorganism application, and the second one is metallic nanoparticle coatings. So I would like to know what are the application of these two innovation, like the fabric resistance toward microbes and the metallic nanoparticle coatings. Is it like what is it used for? Like, is it like in a medicine, like surgical field, or like what are the examples of the use of these innovations? Thank you.
Very good question. I don't know if I mentioned it before, but people working in the food industry, if we can have a lab coat that is antimicrobial, that helps. Most of the contaminations happening in cutting the meat, that kind of events. The cold chain we have, maybe not in the example of the kitchens here, but I mean the university restaurants, but most of the accidents happen by human interfering. So if we can limit this further and further, that helps. If we can have text, of course not for everyday outfit, but in specific environments, not only in hospitals, definitely also in hospitals, the nurses, if they have an antimicrobial jacket, that could help. But people working in the food industry, why not? We can make it in the long term as cheap as possible. The surfaces, I was also mentioning the interest of car manufacturers. The surfaces that we keep microbes free, fantastic. Transfer of COVID. Do you know how it happened? Just touching the door handle. If you can treat the door handle just by a little bit of that coating, why not? There's just a paint, so to say. The rest you will not even see it, but this is where the transfer could happen, right? There are many, many applications. All of in the food industry, all of this is inox, the material. It's perfect coating surface to make it even more resistant to any leftover microorganisms. So I see a lot of possible applications there. Absolutely.
Thank you so much.
Thank you.
Yes.
Anyone would like to ask questions?
No? You have a question?
Gee.
I thought you had.
From our friend again. Okay, please.
Hello, my name is Adam. I'm from Malaysia, the University of Nottingham, Malaysia campus, yeah?
Yes, yes,
Yeah,
yes.
yeah.
Yeah.
First of all, before I begin, I'd like to say thank you so much for the interesting presentation.
Thank you.
I could ask many, many questions, but I'll keep it to one. So I'll build off my friend Mai. I want to ask more about the nanoparticles, because I know they're trending right now, but personally, I think there are many challenges with the nanoparticles, especially in the food industry. For example, I think right now we use a lot of zinc and silver.
Yeah.
Or am I right? Okay. So I'm thinking in terms of producing the nanoparticles and then applying them into food, what exactly are the challenges and how can we overcome these challenges basically?
I don't have a miracle solution, but you're absolutely right, and this is another example of the conservation of misery. We try to fight microorganisms. At the same time, we need very specific raw materials, ingredients, copper, zinc, which is already better than silver. But okay, we still need that material. And materials, rare materials, are now economic power. If we realize how much of these materials are in these things, that's your left hand, right? This phone, the materials now, this is the economic power of different countries. And we make deals with Ukraine because of this. Don't believe it is to stop the war, and that's bullshit. We make deals because we need the materials. We depend on it, so that is a problem. That's absolutely a problem as you sketch it. We should get rid of, now our job as scientists is in the first place to check, does it work, yes or no? At the same time, we are technologists, engineers. We need to see, is it sustainable? And then the answer is sometimes, if not most often, the answer is no. That's why many of these novel developments never reach the stadium of full-scale applications in the industry. What I mentioned to the young lady a minute ago, in that project where we work on these, we, as a team, we check the antimicrobial, antibacterial effect. There's a team in Wageningen who is checking the toxicology effects. These could potentially harm us, and at too large doses, they could even kill you. So we should be very careful, and I'm not at the stage that I would say this is the solution for every problem. I see potential, but it will not solve all the problems that we face in the food industry. Definitely not. And I hesitate to say that they could play a role in packaging. Maybe yes, but definitely not in the eatable packaging. This I don't see happening. That's a bit an initial answer. But toxicology is here the key. And these people, they have models to check what happens when we inhale these particles, what happens and what reaches the lungs and so on. I'm a little bit allergic. I don't know if I have my stuff here with me. Give me 1 second. No, so if I have an attack here, I am in trouble. Let me see. Anyway, you know these puff devices? No. The aerosol, not aerosol, the puff, the spray. The spray. I think so, yeah. It is very interesting to check the fate of the individual particles before they reach the place where you have to go. With Janssen Pharma, we did a study where let me start again. In these devices, you have particles. Can we guarantee that the size of the particles is identical? No. So, that's the same with the nanoparticles that we are studying here ourselves. These nanoparticles, also the ones that we use in drugs, they have different size. The particle size distribution is not a peak, it is a very, yeah? That means the penetration depth into my lungs is what it is, but that depends on the particle size. If I need to reach them, the specific place I want them to be active, I need to overdose, yeah? Because I need that specific size to reach that specific place. And that is the real problem, that we constantly need to overdose rather than put just to have it exactly where we want it. And that was the thing in that study. We tried to reduce, simply said, and that is a milling device they use in the pharma industry, the same mills as we use in powders. We try to reduce the particle size distribution so that we just have to give enough. That's exactly the same with the nanoparticles here. We need to overdose or we are not sure they are effective enough. So we are not yet there. And commercial applications wait a little bit in the food industry, I would say. Toxicology has exactly that to do, and that's what the Dutch colleagues do. Not in humans, of course, but they have that model where they see the inhalation and then see the distribution and the fate of these particles. Half of it stays here. And it goes to your stomach with the saliva and so on. So watch out. But we can keep talking, yes, definitely. Thanks for the question.
Thank you so much.
Of course it does not answer. I don't have the answer. I don't have the answer. We need all of you, the bright people, to solve it step by step.
Yeah, okay.
But keep in mind, solving one problem, you create another one.
Okay. Any more questions, or we have it? Okay. Last questions. Okay.
So, hello everyone. Thank you, Professor. My name is Angela May Perez, all the way from the Philippines, Visayas State University. So I would just like to ask a question. What are the most promising novel technologies to control microbes? For an instant, I am now currently conducting my thesis study, which is evaluating the quality of milk. And it is known that dairy products are highly perishable and vulnerable to microbial spoilage, right? Like coliforms, lactic acid, bacteria, and psychotropes. And freezing and thawing it may stress microorganisms, but still there are survivors that can recover and potentially form biofilms on equipment or packaging surfaces, leading to contamination. So my question is, what are the most promising novel technologies currently approaching commercial application for controlling microbial growth in dairy-based beverages, or maybe not just in dairy-based beverages, but in general? So, Professor or Sir, what do you think are the most promising novel technologies that you can suggest or advise? Thank you.
So if I answer this question, we have solved everything. I should stop the recording. I will charge you first. By the way, I forgot to show this. No. That is a very tough question. And I'm very honest, I don't have the answer. But I do believe that based on experience, seeing what we do with quite a few PhD studies and so on, there is no, well, in the dairy, it is mainly the spores, Bacillus cereus. That is the problem, how to keep them asleep. In another food category, it is another problem. In fresh cut leaves, it is another problem. VTech. We had an outbreak in Spain, a very heavy one with multiple people died. So there is no miracle solution for everything, but coming back again to the answer I gave to the young lady about radio frequency, I do strongly believe, and if you say we should not induce resistance, then my approach is at this moment try to combine technologies. Start from an idea. This is what has been published in the 50s, the 60s, mainly by Dutch people, the hurdle technology. Put different hurdles that you make the life of the bacteria miserable. And these hurdles, you should design them or put them in even the sequence is important, that you attack different mechanisms of resistance. And that's why it is for us as scientists, it's so essential that we know why bacteria resist the treatment we subject them to. Does it help you that you can change the DNA if you cannot change the membrane? No, you will first maybe focus on destroying a little bit of the membrane and then see, can I now go a step beyond, because there will be survivors, and now focus on the DNA, the double-strand DNA, and combine with a more mild heat treatment. Combination of different technologies, to my opinion, is the future. But that will demand a lot of experimental work and a lot of experimental design, because you need not only to identify for your specific target microorganisms the specific technology. What is key here is the sequence of the technologies. And we do now a study with non-thermal plasma and radio frequency. We see differences whether we do this way or this way. And yeah, that would be my first approach. Keep the treatment as mild as possible, and the main trigger in the past has been to safeguard the quality, that we do not destroy vitamins, organoleptic properties, the taste, the crispiness, all of that. The more stringent one, unfortunately, has become the resistance that you build up. If you subject to a variation of technology, you already minimize the risk a little bit. That would be my first approach. I don't have a patent on this.
Okay, sir, that's so informative. Thank you so much.
Thank you. More exam questions for me?
Anyone?
All happy? Then I hope to see you in your second year next year.
Wow. Give him applause.
You all will pass, right? Yeah.
Okay, I think we have enough. Yeah. Yeah. Thank you very much for a very wonderful presentation, and also for all the participants, very engaging conversation and very wonderful question as well. Give us applause, everyone. Wonderful. Yeah. Thank you very much. So I'm handing this session to Mr. Yoga, who is the moderator. Give him applause, everyone, please.
All right, thank you, Jan, Professor Jan. I have said, I don't know, 1,000 thanks maybe to Jan and Monica these days, this week, and also Mr. Haidir. But I don't really think that you understand what was the meaning of Prof. Jan last sentence. I hope to see you on your second year. I don't think that you really get that. It has two meanings. First, of course, Prof. Jan wants you to stay, because there is some people might want to leave after taking first year. This is not for me. The first one is he wants you to stay for the second year and of course until graduation. But the second one, that is even more exciting, means that Prof. Jan is planning to come again next year. Yay! let's hope, yeah, let's hope. But the boss is like, okay, how are you going to fund that?
We'll have a chat.
Okay. But anyway, this is lovely. I know there are more questions coming, but still somebody else, a bigger group is waiting for us. So sadly, we have to end this very exciting and good lecture. I hope you guys learn so much today, yeah? Okay. So thank you so much, and then we close this session, this general lecture. It is recorded, and then it has been broadcasted live, but now it is also, you can see it later on. Yeah, we will share the link. Mr. Haidir, Pak Haidir will send you the link so you can re-watch this beautiful lecture today. So again, with a big applause, yeah, give a big applause for all of us. And yes, thank you so much. I close this session. Thank you again, Mr. Haidir. Wassalamualaikum warahmatullahi wabarakatuh. Thank you. Bye-bye. Thanks for all the help. Thank you so much.
Thank you.
And we have to go.
I think we have to run.
Yeah. You go with me. I'm not driving. This is yours, yeah? Would you like? Oh, you want to?